Hello and welcome to this PIR live event webinar brought to you by Partners in Research Canada. My name is Stacey Joyce and I will be your host. Remember, if you're joining us live, that you can ask questions anytime by clicking Q&A near the top left of your screen. Make sure to let us know where you're joining from or what grade your students are so we can give you a shout out when we ask your question. Today's guest is Dr. Maria DeRosa. She is a professor in the Department of Chemistry at Carleton University in Ontario. And we're going to join her presentation just slightly in progress here. So I hope you enjoy the presentation and the virtual tour.
You probably have at least heard of those three letters, DNA. This is a special molecule inside our bodies that contains our genetic code. What we're looking at here is a DNA double helix. It's actually two molecules of DNA. We've kind of unwound it. And if we take this molecule and unzip it, it's actually two spaghetti-like molecules. They're called like polymers. And you're seeing all these letters show up on the screen. This is the code of DNA, all these G's, C's, T's, A's. This is the code that makes up our genetics. But what we do in nanotechnology is we think about it a different way. We say, imagine that those letters are like bits in a computer, zeros and ones. Or let's imagine that those are a signal or a code for bringing building blocks together. A's and T's coming together, G's and C's coming together. So what we do in our lab is we don't use DNA for genetics, for anything to do really with biology. We use DNA for something called nanotechnology. And nanotechnology is basically just the science and technology of the very small, billionths of a meter. So if I took a meter stick and chopped it up into a billion pieces, it's really hard to imagine that, but that's how big a nanometer would be. Nanotechnology works on the scale of 0 or 1 to 100 nanometers, so very, very small. But DNA is actually a perfect building block for working on that length scale. So on one side of your computer screen, you should be able to see the double helix. And the double helix of DNA, those two molecules all wound up together, when they're set up like this, it's about 2.5 nanometers wide. And about 10 of these stacks on this base paired double helix, on these different rungs of the ladder, is about 3.5 nanometers. So the dimensions of DNA are perfect for nanotechnology. The other thing that's perfect about DNA as a building block, as a synthetic thing that we can use to build new molecules, new materials, is the fact that we can make DNA in the lab. So on your left-hand side, you see a DNA synthesizer. I'm going to show you one of those in our lab, and I'll show you a little bit about how it works. But on this instrument, we can actually make DNA. So you may have done in elementary school or high school experiments to extract DNA out of, onion skin or out of a cheek swab. We don't need to do that to make our nanotechnology. We build DNA bit by bit on one of these synthesizers, and I'm going to show you how that works in a minute. So then what do we do with this DNA? What's special about the DNA that we're building on our synthesizers? We're making a type of DNA called an aptamer. And an aptamer, that apta part, it comes from Greek fitting or to fit. These are synthetic pieces of DNA that fit around a molecule or a target. So you can see on the left-hand side, there's a red squiggle, right? And this squiggle is one of the two strands that make up the DNA double helix. So it's one strand of DNA. And it's folded up onto itself to make its own structure. And at the top, you see there's a blue, that's another molecule, a blue molecule that is fitting into like a little pocket that DNA squiggle has made. So that fitting of the DNA around a target molecule, that's like a recognition event. This piece of DNA recognizes and sticks to this molecule, this blue molecule, and no other molecule. So it makes it a really good building block for like a sensor. So you can sort of think of it like the idea of a lock and a key, okay? So if the lock is this piece of DNA, this aptamer, and the key is a molecule I want to detect. So I want to know, is this molecule X in my food? Is it in my water? I could get a lock that recognizes only that key, only that molecule. So in our lab, we develop these aptamers that are locks, essentially, for all sorts of different molecular keys. Molecules that could be present in anywhere in the world around us that we want to be able to detect and have an idea how much of this is around. So what you're going to see now is a little dance. So this came from the students in my lab. They entered a contest called Dance Your PhD. It was many years ago now. But they danced the process that we do in the lab that we use to find these aptamers, find these molecules that can recognize other molecules. Here you're just seeing a tiny little snippet, but what you're seeing is you see one young lady with a target on her shirt. She's supposed to represent the molecule we want to detect. And everyone else around who's dancing, those are all DNA. And the idea with finding aptamers is that we expose a target molecule to lots and lots of different types of DNA. We keep the ones that stick and we discard the ones that don't. So in this video, the idea was the DNA that dances the same as the target, we keep, and everybody else dances away. We get rid of them. And so the students actually ended up winning Dance Your PhD that year, and they got to go to New York, and they won $1,000, so very proud of them. But I use this video to explain a little bit in simple ways what we are actually doing in our lab. So I'll show you a little bit about that when we actually go into the lab and look around. Okay, so in our lab, we make these aptamers, and they detect molecules. They stick to molecules, so we can say, hey, molecule X is in your food or in your water or whatever we're looking at, in your blood. So now I'm going to ask you a question. Did you eat any of these things for breakfast this morning? Did you have some kind of cornflakes or oats, peanut butter on toast? Did you drink coffee? Any of those things are, food and drink. We may be interested to know what's inside these things. Is there any molecules that we should be concerned about? So it turns out that there is a molecule or type of molecule we're interested in detecting in all these different types of food and drink. And these are called mycotoxins. Okay? Not to alarm you, but these toxins are present in our food. They're natural toxins. So there are natural ways that they're produced that ends up in our food. So Canada, for example, does a great job managing these toxins and making sure that we don't ever get exposed to a lot of them. But the way that they do that is by doing lots and lots of testing of the grains and the materials that make up our food. So here's a picture of a number of trucks lined up full of grains. They're coming from farms and they're going to a grain elevator. And somebody you can't even see at the end of this picture is a grain elevator, and there's somebody there who's testing the grains to see if these toxins are present. It takes a while. So what this picture is supposed to represent is that in my lab, I want to use aptamers to make the tests that we have to do on our food faster, easier, cheaper, so that your food is cheaper and the people that are able to do these tests can do them quickly so they can get that food out to people who need it right away. So what I'm going to show you in a couple minutes is how we make these easy tests that detect these toxins, that use DNA to be able to give us that yes, this is present, or no, it's not. So these little tests that I'm going to show you are called lateral flow assays. And I don't know if you've ever seen one of these things before, but if you've ever seen a pregnancy test, this is a lateral flow assay that you could buy at the drugstore. We make similar ones, except that we want to use them on a farm or at a grain elevator. And the way they work is that aptamers inside them will stick to the molecule of interest, and if it's present, a dot will disappear. So we will see a dot if there's a very little, and then it will disappear. So as we can use this in our, just an easy yes, no, is there a toxin present? That's something that anybody even without any science background will be able to see, yes, I see a dot, or no, I don't. So I'm going to show you those in action. Okay, so before I actually take you into the lab and show you around, I just want to say that I'm going to show you another thing that's really important about the work that I do, and that is that we have lots of students who are actually the ones doing the work. So here's a picture of a recent picture from my lab group, all these different students and postdoctoral fellows and research assistants who work in my lab and do this job of finding aptamers and making tests out of them. So you'll meet a few people in the lab today as we go through the lab, and they're happy to answer questions as well as I am. So at this point, I can answer a few questions about the presentation if you like, or we can move right away into the lab and take a look around.
Well, I guess I can chime in here with one question about aptamers. You mentioned the mycotoxins as one application. What other applications do you have that you're working on, or do you see as the future for aptamers?
Great question. So mycotoxins, so toxins in food are just one aspect of our work. We're very interested in understanding things that are outside of food. So we have aptamers that detect neurotransmitters. So these are chemicals in our brains that help us function. We have aptamers that detect the protein that's involved in Parkinson's. So we want to understand health-related questions, diseases, using aptamers. We have aptamers that are used in coatings, so that when they recognize whatever their molecule is of interest, they release whatever's in the coating. So this could be useful for drug delivery, like I want to treat a cancer cell and not a healthy cell. So the aptamer recognizes cancer cell and then it releases the drug. So we are interested in health problems, environment problems, agriculture, all sorts of things, because aptamers are very general. They can be selected to bind almost anything.
Wow. And we tend to think of chemistry as this narrow field, but that's a lot of different applications that you can reach with this one type of nanotechnology. So perfect. I'm going to then, I'm going to bridge the gap here, and we'll go into the lab. And Dr. DeRosa is going to literally run across the hall to the other room. All right, sounds good. So we'll mute that first computer. We'll unmute our lab computer. And if we can get our undergraduate student, Evan, to say hi there.
Hey there.
There he is. Perfect. Maybe you can let us know your role in the lab, Evan.
So this summer I'm in Maria's lab, just doing my research project and synthesizing DNA.
Just a lot of synthesizing DNA. Awesome. Fun. So how many years of university have you put in so far before this summer?
So far, 3 years.
There we go. So, and your graduate students, Maria, how many years of post-secondary education have they usually put in so far?
So it depends. So we have people like Evan who are undergraduates. We have people who are master's students. You'll meet one in the other lab. Dan is a master's student. So he'd have done 4 years of undergrad, and he'll be working on 2 years of master's, so that adds up 6. And then we have other students who are PhD students. They'll have another 2, 3, 4, 5 years added onto that. And then we have other people called postdoctoral fellows, and those people have completed their PhDs. Maybe they've done 10 years of post-secondary or more, and they're still continuing to work in labs and expand their skill set before they would take on a job like what I have as a associate professor.
Awesome. Well, so a little bit in our transition there about how you maybe get to these places, but I'll let you take it away again, Maria. You can tell us what it is we're looking at.
Perfect. So buzzing away in the background, I'm going to actually bring the tablet a little bit closer in a minute, but buzzing away in the background, this is the DNA synthesizer that I talked about in my presentation. So this is an amazing instrument that allows us to build DNA base by base, so little bit by little bit. I showed you that little animation with A's, G's, C's, and T's making up this long spaghetti molecule. This is making that spaghetti molecule, and they're making it on a small glass bead. Small glass bead, base by base, we're getting these little spaghetti strings that are getting formed in real time. And what's nice about this is that we can control what's actually getting built. So Evan, what he's done is pulled out these little containers that contain the beads. I think I'll bring the tablet a little bit closer, so you hold that. So you won't be able to see me for a little while, but I think that's a good break. So I'm bringing you close to Evan. So what he's showing you, these little, plastic containers, they contain little beads. There's a white powder that's at the bottom of them. And that's where we start building DNA. So on those little beads, they're micro-sized beads. You can't really see them. Yep. They contain the first base, so say a G or a T or an A.
These ones happen to be C.
Those are C bases. Wonderful. So what we would do is that's going to be the first base in our sequence. And because, like I said, unlike in biology, we would extract DNA and it would be the sequence that it is for whatever natural reason. On our synthesizer, you can see there's a computer there. We could program in exactly the sequence we want. So if in another experiment, Evan has figured out that, well, the aptamer that binds to a dopamine molecule, which is a neurotransmitter, has a sequence of G, A, C, 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 whatever, then we can program that into the synthesizer and build it. So I'm going to bring you a little bit closer to the actual synthesizer. You can see it buzzing away. So as you're looking inside this top port, you can see that we've got this this one section that it'll come back and forth, and every once in a while you'll see there are those beads right in the little containers, right? That's going to move that back and forth, this chuck. And what's happening is that up here, you can see these bottles. These bottles contain those building blocks, the A's, the G's, the C's, and T's. And according to whatever we've programmed, they're getting injected one at a time, you know, for a certain period of time into those little containers. And eventually when we're all done, we'll be able to take them out. And oh, you can see actually Evan's opening it up for us a little bit. So out of each of those little ports, let me move it this way. There we go. You can see those little, there you go. That's where we're injecting all these different bases. We can even add in bases that aren't natural at all. We can make totally modified bases that get stuck onto the DNA. Say if we want to make something that glows or something that is a color, then we can add that on using the DNA synthesizer, which you can't make biologically. So when it's all done, we would chop off using some chemistry the DNA off the beads, and then we would be able to take that and use it in an application. So that's what I'm going to show you next. So I'm just going to say goodbye to Evan. Bye, Evan. I'm going to put this back down, and we're going to move over to the other lab, and Dan and Velu are waiting for you, and I'll be over there in a minute.
Perfect. Okay, thank you so much, Evan. So I'm going to mute that surface right now, and we will unmute those who are in our secondary lab here. Hi, guys. Thanks for showing us around. Maybe you could introduce yourselves one at a time while we're waiting for Maria to walk down the hall.
Sure. My name is Daniel, and I'm currently finishing up my master's, so that's a 2-year program on top of my undergrad. And this year is Dr. Velu.
Hi, I'm Velu Ranganathan. I'm doing postdoc here since 2012. Same working outer sensor and various colorimetric and fluorometric sensor tube, everything. Hi.
Thank you.
I'm Satya. I'm doing here as a visiting student researcher. I'm continuing my PhD research in the Maria's labs. Same assay-based microbes detections I'm doing.
Well, thank you so much for introducing yourselves. I think we've got Dr. DeRosa there as well. Now, what I'm just seeing from our laptop here, should I switch over to the surface?
So yeah, if you want to switch to the surface, then I can come closer to the instrument that Dan's going to show us, which is our printer that prints those little strips tests that I was showing you in our presentation. So I'm going to turn to this.
Okay, so give me one moment here. I'm going to mute that laptop and unmute your surface so we can see. Go ahead and say something to the surface and it'll switch our video over.
Go ahead, Dan.
Okay.
So I've just muted them both for a minute. We're going to have to turn the volume down on the laptop, I think. Or turn the microphone off on the laptop.
It's actually a assembly line, which includes a laminator, printer, and cutting device. And they're all specifically made for printing lateral flow devices. So these are the kind of pregnancy strips type assays that Maria had showed you previously in her PowerPoint presentation. So what we have here is a nitrocellulose membrane or some sort of paper backing in which we would print a fluorescent DNA label that would be selected to bind a certain target. So in our case, we would be looking at food toxins.
That's right.
So what I'm going to do now is I'm going to start the printer, and you guys will get a chance to see kind of how it deposits the DNA onto the paper backing that we have. So it'll do 2 runs. The first one, it'll print a line of our first DNA recognition element, and then the second time, I'll allow the printer to pass, and it'll deposit dots. So I'll start the machine, and you guys will get to see how it works.
So while you're doing that as well, Dan, can you point to where the DNA is and that which vials contain the DNA and that kind of stuff?
Yeah,
too?
sure.
Sure,
So,
you can start it while you do
okay.
that. Yeah, go for it. It's going. It's filling. That's okay. So you can see that there's a spot moving. I think I might move a bit closer so you get a better idea. I don't know how well you can see that, but basically you can see there's paper, and it just drew a line. Now the line is faint, but that line is what contains our DNA. Now it's making spots. Let's see if we can. Once it's done, I'll see if I can get a little closer to it and we can. Maybe Stacy can give me a heads up. Can you see that, Stacy? dots
a little,
and lines to a certain extent.
Yeah, if you back up just a couple inches, then the shadow will come off. There we go. Not sure if you can hear me now.
I can hear you.
Yeah, I think we can see those dots. They're faint, but I can see them.
So they're fluorescently labeled DNA. So we wouldn't necessarily see these ones by eye. I'm just going to move over to the vial. So maybe with you can point to them because my hand's back here. The two contain, yeah, inside here is where we have our DNA that's getting deposited. So once we've kind of made these strips, right, we would chop them up, right? I don't know if Dan's going to show us how that works.
Yep.
Okay, great. Chop them up into these little tests that are going to get assembled, and then Vailu's going to show you what they look like once they're all assembled.
So this instrument here is automatically chopping that long bit into short strips. Do I have that?
Exactly. It's going to cut it up into all sorts of smaller strips. Then I'm going to come over around. You see, they're getting tossed out there, right? So we used to do all this by hand. You can imagine it was like arts and crafts. It was a bit tricky. So then now what ends up happening, I'm going to walk over like a cooking show. You're going to see this is what it looks like when they're all assembled. So I'm going to bring it a little closer. So what you're looking at are these strip tests, okay? And you can see, I guess it's also in reverse because of the way the camera is working, but there's C and T, basically control and test. And you can see that they have different red dots on them. So depending, some of these are completely assembled. Like I'm going to pick one up. This one, maybe Vailu can hold that one up. This one is like completely assembled. Let's see if it will focus in on it. And so this is what this kind of looks like I said, one of these mini pregnancy tests. And you can see dots. When you see the red dots, that can mean yes. Yeah. And if when you lose one of those dots, oops, I'm losing the spot here. There we go. Perfect. Then you know that it's an easy yes, no answer based on the color. Taking that apart, then we can sort of see them. Actually, leave this one down there for a second, Vailu. Yeah. I just want to show these other ones. So these ones here that we've taken off the, I have to figure it out. There we go. Those ones beside it, we've taken off that top casing so you can see what they look like. Those little strips that jumped out of the printer and got chopped up, that's where they are there. So this is just a way for us to make these really inexpensive, I'm going to just zoom in on Vailu because he's the one who does all this work. These really cheap but effective tests that are really easy for just anybody. A farmer could use this, be able to detect if these toxins are present in their food. Or, for example, like I said, we could use them for health applications. So that's the end of the tour. If we're okay with that, then I'll go back and sit at the other computer in the other room and we can answer questions.
Perfect.
Okay, so let me just put this down. You guys can just take a last look at the lab. Everyone say goodbye while I go back.
Lots of waving hands. Thanks so much for showing us around the lab, everyone. That was excellent.
You're welcome.
Okay.
All right, so we've got the lab muted and we have Dr. DeRosa sitting ready for questions. So make sure if you have any questions, don't delay. Just type those in as you have them and we'll relay them to Dr. DeRosa in turn. So I guess we talked a bit already about your path to becoming a researcher and a group leader as well as a professor. But what do you think is for you the most challenging part of this career?
That's a great question. So I'll first tell you what I love because it's always better to think about bad stuff. So I love working in the lab with the students, mentoring the students and helping them kind of find their path. But in order to do that, I have to get resources, I have to get funding so that we have that equipment, that we have the chemicals, the students get paid, these kinds of things. So one of the hardest parts of my job is that I have to write grant applications and write up all the work that we're doing so that people understand that it's important and then will be willing to support it, right? So at the end of my presentation, you saw the picture of the lab group and then there was all these different other supporting organizations that I wanted to thank that were on my acknowledgement slide. All those people help us do what we do in that lab. They give us funding. So you can imagine that it's another full-time job to coordinate all those people and to get all that funding and make sure that we're delivering everything we promise we deliver and that kind of thing. So the administration of the lab is actually a really big job. and it takes me away from the lab, which is hard, right? But I do it because I love what we're able to accomplish in the lab, and I love working with those students, so it's all worthwhile. And all those supporting organizations, honestly, without them, you know, we wouldn't be able to do any of this stuff. So we're so grateful for whatever they can provide us, and it's worth the effort to reach out to them and to help them understand what we're doing.
Awesome. We have a question coming in from the grade 6s and 7s in Sault Ste. Marie. They want to know how exactly you use those strips to test food.
Great question. So it depends what the food is, right? So if it's a drink, it's a lot easier. We could just dip that strip in or put a drop on top of the strip. If it's a solid food, so a lot of what we're interested in are grains. You saw like cereal, you know, cornflakes, this kind of stuff. Then what would have to happen is basically a sample of that grain would just have to get mushed up. Doesn't have to be in any fancy equipment, just basically crushed up, water added, and then the water gets, a drop of that ends up on the strip. So in, you know, the pharmacy, when you see these strips, what ends up happening is you have to use urine, right? It's a urine test. So for us, we would use some kind of liquid, usually water. So if it's a liquid already, we just test directly. If it's not, we just have to take a little bit of our sample and put it in water and then test on it. Good question.
Great. Well, and thank you so much to the students in Sault Ste. Marie for that question. Keep them coming. We still have a couple more minutes here. So we have a question coming from Southern Ontario asking, do you ever have high school students in your lab?
Great question, actually all the time. So every year we have at least one, sometimes two high school students who will work during the year towards a science fair project. So, you know, you can imagine a lot of high schools do science fairs. You don't have to do those science fairs just in your kitchen. A lot of students now are coming into real research labs and asking questions that they want to have answered for their science fair, and we work with them and the equipment that we have and try to help them answer their science fair questions. So that's a lot of fun, actually, and then those students get a lot of interesting training that ends up a lot of time making them want to go into science careers. So we definitely take high school students, and you know, you should check it out in whatever universities are closest to you. A lot of them will take you on for a science fair project, so go for it.
Great. And one of our final questions, at least, is what types of other researchers or other personnel do you find yourself collaborating with along the way?
Great question. So, you know, we are chemists and we work with this special chemical, this material, which is our DNA. But, you know, that can only take us so far to solving the problems we want to solve, to answering the questions we want to answer. So a lot of times we have to collaborate. We have to work with other scientists and engineers. So, you know, the simple tests that we make, they're easy to make, but they're, you know, they're a yes/no test. If I want to make a really fancy sensor that could, say, work remotely, you know, it might monitor the field with drones or something like this. You can imagine there's this idea out there of precision agriculture. We have to work with engineers who understand these types of radio frequency sensors that we would love to use. So in that case, engineers are who we work with. On our health side, when we're trying to find aptamers and make sensors that detect health-related targets, like cancer cells or neurotransmitters, in those cases, we need to work with health researchers. We need to work with neuroscientists, and we do. We need to work with cancer biologists because they're the experts in that area, and all we can do is provide them a tool. Hey, we found an aptamer that could be useful to you, but they have to show us, you know, what their needs are and how we can all work together.
Wow. I do have one last question getting in here under the wire from Kendra in the Sioux. She wants to know, do you use those strips to test water in particular?
Good question. So right now we're not, but there is a project ongoing that we're going in that direction because we have aptamers that detect different viruses and bacteria that could be in water. So you can imagine people want to know, is there E. coli in my water? Is there norovirus in my water? All sorts of these organisms that could make us sick could be in our water supply, and we do routinely test for it. So this would be a cheaper, easier way to test for it, right? And potentially even something you could do at home one day if we were able to do that. So definitely we're interested in testing water. And water is actually a wonderful medium for our tests. It actually works really well. It's easier than, say, testing corn or testing blood if you're doing a health test. So I think there's a lot of promise in that direction for sure.
Great question, Kendra. Thank you so much for that. So my very last question for the day is if you have any advice or words of wisdom for students who may have thought all of this was really neat today and it's something that they want to pursue, what would you want them to know?
Amazing. So what I would say is that when I was a student, when I was in high school or elementary school, because I know they're both listening, I had no idea about any of this stuff, right? I had the job I'm doing today, it didn't really even exist when I was in school, right? So this idea of working with DNA and nanotechnology, none of it even existed. So how do you prepare for a career that doesn't exist yet? It's really hard to do that. So what I suggest is keep an open mind, stay in science and math, because those fields help you to answer questions. And new questions will emerge, and new technology, new fields will emerge, right? But if you have the skills on how to ask and answer questions, then you could face anything, right? So if this particular field interests you, then stay biology, chemistry, math, all those things, and definitely you could do something like this. But there's a good chance that by the time you get here to this age, there's going to be a whole other fields that have opened up that are doing amazing things, right? So if you want to be ready for that, then you have to stay curious. You have to just, try your best with the idea of keeping up with science and using that as a tool to answer questions for all different fields.
Well, thank you so much for taking the time today, showing us around your lab, showing us what it is that you're doing, why it's important, and answering our student questions today.
My pleasure.
So just a quick note that next up on PIR Live event, we are getting the first look at MindFuel's new Canada 150 video game, celebrating Canada's inventions over the years. More information on this and other upcoming webinars is available at pirweb.org. Thanks so much for tuning in, everyone. We'll see you next time.